recombination should sound familiar to you, even though we haven't discussed exactly how it occurs outside of its product of crossing over. So homologous recombination is a process where DNA segments that are similar or identical exchange information. They break and rejoin to give you a new combination or recombination. So this can occur in chromatids. So sister chromatids, we know they are identical. So if they exchange genetic information, it's called sister chromatid exchange, but there's really been no change because they are identical. But if homologous chromosomes cross over, now you have a genetic recombination because they are similar, same genes but different alleles. So you end up with recombinant offspring, recombinant gametes or progeny that we talked about when we talked about genetic mapping and linkage. So this is important because this gives us genetic diversity. And we've talked about that time and time again with meiosis. One of the number one reasons to do it is to introduce variability into the population. Another reason is DNA repair. So because we have homologs or two similar chromosomes, if something happens to one out of the pair, the other one is utilized to rebuild it. I also want you to make note that this is a highly regulated process and there are proteins that play a role. These proteins tend to be called REC proteins. So if you see REC, which is how recombination starts. So here's our REC proteins. So there are two models proposed to allow for genetic homologous recombination to occur, the Holiday model and the double-strand model. So the Holiday model says that we have two chromatids, or homologous chromatids, are nicked. So both molecules each get a single cut. And after you get that cut, they link to each other. So this is where they cross over, and this is a Holiday junction. This Holiday junction mitigates. So now it starts to go in and try to figure out what's going on here. It's like they are literally giving one piece and taking another. So the more I give, the more I take. And so I'm leaving an area that's about the similar size in both of them. And this is a heteroduplex, hetero because they are different and they are combined. So we have two here together. Now what's going on is this bending. So remember, we still have two arms on our chromosomes. So we have the one that was nicked and crossing over, and then we have the one that wasn't. And so and then you have to resolve this. So you have to cut again to separate the duplex. And you end up with one arm that hasn't been touched or recombined and one arm that has. And this introduces some variability. The other model is a double-strand break model, and this is saying that one arm gets two breaks. So we're going to have a double-stranded break here. Now this is probably widely recognized as being an error, and so we start to repair it, and in doing so, we cut away a part of this. And when we get rid of that material, it's on both strands, so we can't figure out what's supposed to be there. We have to rely on the intact homolog. When we go to this homolog, it's similar, not identical. So we form this D loop, and in this D loop, we are using one side of this DNA as a template to rebuild what's missing here. And in doing such, since they are similar, not identical, we are changing essentially the sequence here, okay? And so now you end up with a recombined chromosome. Now, I'm less interested in you knowing exactly how they're done and more interested in you understanding the importance of it. So as far as the process, understand that the DNA has to be broken. Okay, so we are introducing a double-stranded or a nick into the DNA. Understand that the homolog is used as a template for the broken strand in the double-stranded molecule or model, and in the Holiday that each strand is being used to rebuild the other, and that the end result is recombination. And the importance of this can lend towards gene conversion. So what happens in gene conversion? And it should sound like the name, we are converting a gene. So here we have our Holiday junction and this branch migration. So it's, essentially invading the other strand. And you get these two heteroduplexes, and now they are different here, where this arm has a G and this one has an A. So this is a mismatch repair. Now we have to come in and correct that. Well, there are a couple ways you can correct. So in the first one, and remember, strand 1, strand 2, strand 1, strand 2. So when we correct it, we ultimately make strand 2 resemble strand 1, so we have gene conversion. Whereas here, strand 1 looks like strand 1, strand 2 is strand 2. There's no gene conversion. Here, we change gene 1 to have, here we make, this is gene 1, 2. They're switched, but there's no gene conversion. And in here, they both resemble gene 2. So let's slow down and go again. So if we have 1 and 2, and this is 1 and 2. So here, 1 is still 1, 2 looks like 1. Here, 1 is still 1, 2 looks like 2. Here, 1 is 2, and 2 is 1, so there's no change. And here, 1 is 2, and 2 is 2, so we have converted it. Okay. Then there's the double-stranded model that also allows for gene conversion. So here we have a recessive allele B and a dominant B. We get this double-stranded break. We degrade the DNA surrounding the break. We use the dominant strand to correct the break, and by doing so, we essentially replicate that dominant strand, inserting a dominant where a recessive used to be, thereby converting that gene. Okay, so I want you to take away the larger picture here of what recombination is, that the REC proteins are involved, that it's important for gene conversion and DNA repair, that when homologous recombination occurs, you have to have some kind of break and then some kind of repair, and just take away big pictures. Don't get bogged down with, oh, this happens and that happens, but what happens and why it happens. So a gene is converted because you're using a homolog as opposed to an identical sister, and therefore you introduced a new sequence. Okay, I hope this makes some kind of sense to you.